So we're going to talk about bacterial habitat today. So with bacteria habitat, we are going to look at where they're actually going to live. So bacteria live in places that allow for them to eat. If they cannot eat, they can't reproduce. If they can't reproduce, well, they have no point in life. And so we have a few different situations, and you can see over here I have some test tubes imaging that. So the test tube is just full of a nutrient-rich media that allows for the bacteria to kind of grow and live in there. So the first one is obligated anaerobes. So these guys will be found at the top of the bacteria. They have to have oxygen present in order to eat. So they are obligated to be in that oxygen-rich environment. We are also obligated anaerobes. We have to have oxygen, otherwise we die. The second one is aerobic anaerobes. They do not need oxygen to grow, and so we see them in test tube B. They're sitting at the bottom. They go through processes like fermentation. They don't necessarily need oxygen, or they can live if oxygen's present, but they don't necessarily need it. We'll have our facilitated anaerobes, which are at the bottom, but it is our test tube C. These guys will go through anaerobic respiration and aerobic respiration. So they can live in both. If they have to go through anaerobic respiration, they will go through what we call glycolysis and fermentation. And then we have our obligated anaerobes. So these guys will die if they're exposed to oxygen, and they do not survive. So atmospheric oxygen, we don't see them on the surface necessarily. These are what we think would be bacteria that we would find on a planet such as Mars. But for us, we find them within the Earth's surface. And so these guys are living down below the Earth's atmospheric surface in the geological site. And so we have different temperatures in which bacteria will grow. So not only do they have to worry about their food source, but also the temperatures. And so we will usually find most bacteria are mesophils. They grow best between that 25 to 40 degrees Celsius. This is 77 to 104 degrees Fahrenheit. Those are where we find our mesophils. That's normal bacteria that lives on your body. And then we have our thermophils. Our thermophils will live in relatively high conditions, and so they like to be above 113 degrees Fahrenheit or 45 degrees Celsius. We do have some hyperthermophils. They live at really extreme temperatures, like we're talking over that 70 degrees Celsius to 110. So we're looking at over 150 degrees Celsius, so what we would find in a volcano. And then our normal thermophils will be found in things like a geyser, like in Yosemite. So we also have our psychophils. These are found in very, very cold conditions. They live in the Arctic. We find them in ice, so they can survive in these extremely low temperatures. So these are a relatively new species of bacteria because not a lot of people have explored the Arctic. And so we do know that there is a lake in the Arctic that does have these type of bacteria living in there and doing very, very well. Bacteria will reproduce as well. So once they have their food and they have a great living habitat, they will reproduce. Usually they'll reproduce during binary fission. Binary fission is a type of asexual reproduction. This can happen in a very short period of time. So if we would actually be doing bacterial collection, we would take a swab or a sample, and within 24 hours, you would have enough on your petri dish to visibly see them. That's hundreds of thousands of bacteria in one little dish. And so binary fission can happen in a few hours, especially if they're if they have nutrients available to them. So remember, if they are aerobic bacteria, they need to have oxygen. If they're anaerobic, you would take the oxygen away. And then they have the temperatures in which they do best in. Most are mesophilic, so they'll be in this room temperature area. Once they're in that optimal condition, binary fission can happen extremely fast. Remember, they're just going through mitosis and splitting. And so they're exact replicas of each other for this. Another thing that's really cool is that we can go through very easily a genetic transfer or genetic recombination or bacterial conjunction, however you want to say it. It's all the same thing. Bacteria has these chromosomes and they also have a plasmid. We can actually take this plasmid and remove it and put it somewhere else. And so the picture that you see onto your right is actually taking this mobile plasmid, and notice how we're taking this pulse and we're moving or recombinating the genetic information. So at the end, they both have this plasmid. We can transfer a plasmid pretty easily, and it's just taking a segment of DNA and exchanging it or introducing it to a new bacteria. This can be really helpful for creating gene therapies. However, it can be also very devastating when we talk about creating a new form of bacteria that could cause an unknown disease. So this could be genetic warfare as well. So we have to be very careful when we're starting to look at this, but we are looking at recombination to actually use as a gene therapy so that we can introduce a bacteria into people so that they can produce a protein that we don't normally produce or our body stopped producing. So let's talk about bacteria in humans now. So bacteria within us is normal. We have a symbiotic relationship with bacteria. In fact, we have a ton of bacteria all over us. Like I stated before, we have so much bacteria inside of us. It is amazing. And we have probably more bacteria than we do cells. It's crazy how much bacteria we have. And so it's really important that we understand that this relationship is necessary for us. We need it within our nose, our stomach, our mouth, our gut, our skin, our vagina, everywhere. And so it keeps us healthy. This microculture that's within inside of us allows for us to be a stable, healthy person. However, if we would happen to have this microculture go off a little bit, say you don't brush your teeth for the next 10 years, your mouth is going to probably become a little bit of a rotted mess and your breath is going to stink really bad. If you don't eat healthy, your gut is going to just go crazy and you're going to have problems, gastrointestinal problems, where you're not going to be able to digest certain things very well and you're going to have stomach cramping. In your stomach, if you don't take care of that, you can have pylori bacteria in there that can cause ulcers, acid reflux, and most people don't even know that they have it. So you're going to really take care of this stuff. And that's by eating healthy, drinking lots of fluids, and really kind of having good hygiene. And so we can also use bacteria to create a lot of products, such as ethanol, such as all of our ethanol plants around here. We use it for drug production to actually make vaccines, especially now that people have egg allergies. We can actually introduce, through genetic recombination, certain segments of antibodies into them, and then they'll produce them, and then we can use them to make vaccines. We use them to clean up oil spill and toxic waste. Sorry for the spelling error. For killing pests associated with plants, and gene therapies. So like I said before, we can now recombinate this bacteria to form something crazy and new so that we can introduce it into the body so it can actually create proteins that our bodies can no longer create. We can also use it to ferment foods, and fermentation, when you think about it, sounds kind of disgusting, but I love cheese, and cheese is just fermented milk. So there's a lot of products that we use in foods, like cottage cheese, almost every fermented dairy product. I love cheese a lot. Yogurts, those are all my favorite things. And those have natural bacteria that ferment them. When you're older, in the age of 21, you can use it to things like wine. That's a fermentation as well. But let's talk about something that's really important, and that's diseases and controlling the spread of this bacteria. So we just said that bacteria is healthy and all over us, but not all bacteria is good for us. Bacteria can spread through touching, coughing, sneezing on our food, water, and other objects. What does that mean? So if I have strep throat, I probably am spreading some type of streptococcus around, and then I'm touching my hand to somebody else's hand, and then I'm putting it near my face or my mouth. And then I get strep throat, which we can take antibiotics for later on to kill. If I have things in my food like salmonella, I would eat that, and then I would get not so good food poisoning. I can drink things, bacteria that is in the water that can make me really sick. Anything like that, that can be a really big problem. So we need to look about how these things are being spread. Sometimes they're spread by a vector, and I'm sure you've heard the word vector a lot lately in the news. A vector is a host or a carrier of a disease, such as a tick can spread Lyme disease to you. The vector or host does not get infected by it, but it is kind of like they're transmitting it to something else. A vector can spread any pathogen. So a bacterium, a protist, or a virus can be spread via a vector. The vector does not necessarily have or show signs and symptoms of that infection. They're just transmitting that infection to other people or other things. So how can we stop the spread of these? One, we can cook our food properly to stop things like salmonella. Boil water before you drink it if you get it from a surface. Wash your hands a lot. Take showers. Hygiene is good. Brush your teeth. Okay, and then we can clean our surfaces. We can use soap and water. We can use chlorine bleach. Those are all disinfectants. We should be cleanly, especially in this time. And then once we have it, such as like strep throat, we want to take antibiotics to remove it from our system. If you're given 10 days worth of antibiotics, you need to take all 10 days worth of antibiotics. If you don't take all of your antibiotics, you're actually creating an antibiotic-resistant bacteria, and this can be devastating because now the normal antibiotics don't work to cure this disease, and you have to now have another form of an antibiotic to cure it. The problem is that now we have antibiotic resistance, and we don't have drugs to cure those diseases, and they can cause a lot of problems later on. So let's talk about viruses. Viruses are crazy. So viruses, what is a virus? A virus is essentially genetic material surrounded by a protein capsule. It's not like a prokaryote or eukaryote because a virus can't go through metabolic processes. It just has this protein capsid. Viruses are not technically alive because viruses cannot reproduce. So basically what a virus does, like this little beautiful bacteriophage here, it inserts its genetic material into a cell. And that cell will do one of two things. It will reproduce until it's so full of viruses that it explodes, or it reproduces with the virus's genetic material into it. We'll talk about viral reproduction here in a little bit. But viruses aren't alive. They're not able to maintain homeostasis, and they're not able to reproduce on their own. So metabolic processes are out the window. Here's the problem with viruses. Viruses can mutate every time they have a new host. So it's really hard to say that one vaccination is going to cure a virus, or when we're starting to make antibodies, we have to make them a viral antibody for a large population of people. So everybody's genetic information is different, so a virus will change. Hence why influenza, every year the flu shot, it's a guessing game because there are more than one strands of influenza, and the influenza strands do mutate as they go into one host to the next. So as we get our DNA mixed with viral DNA, the virus that comes out is a little genetically different than the virus that came in. So do remember that viruses are a little bit different. And this is the end of part